friends, I am Antika and in this video I will be discussing about the sexual reproduction in Chara. I have made earlier videos where I have dealt with the vegetative reproduction in Chara, and also another video in which I have discussed about the general overview of Chara, its thallus organization, its ecological, economic as well as research importance of Chara. Here in this video, I will be describing about the male and the female reproductive structure, the process of fertilization and the germination of ooth spore in Chara. So let's start the video. Chara is a very important species because it is present in both MSc and BSc syllabus. This Chara, I have made videos in parts about various topics in Chara. So in this topic, we are going to discuss about the sexual reproduction in Chara. Now, Chara we should know that Chara, though cosmopolitan in distribution, it prefers very low temperature. That is why it is mostly concentrated in the temperate region. And out of 180 species in Chara, in India we have just 28 species. And Chara is a very important algae because it is an ecological indicator, because Chara, it can grow in alkaline as well as saline water also, but it cannot grow in eutrophied water. It cannot grow in dirty water. So if there is Chara in a water body, we can say that water body is quite has low impurities. I mean, the water is clear. So this is a good ecological indicator species. So sexual reproduction in Chara is mainly oogamous type of reproduction. And the sex organ, they are born at the nodes of the branches of limited growth or the primary lateral. Here we see these are the primary lateral. We know that the Chara has a central axis which is divided into node and internode, and from the node arises the primary lateral or the branches of limited growth. And this is the zoomed view of the primary lateral. Here we see at the nodes the structures, that is the male reproductive structure which is called the globule, and the female reproductive structure which is called the nucleus. They are born at the nodes at the axil of stipules. See this one is the stipule, and this is the nucleus, this is the globule, and this is the stipules. So the reproductive organs in Chara, the sexual reproductive organ in Chara, they are born at the node of the primary laterals and at the axil of the stipules. Primary lateral, the axil of the stipule, at the node portion, this reproduction occurs, and this reproductive organs occur, and the reproduction is oogamy, which means that not only the fusing gametes are different physiologically, chemically, but also the organ that is producing this the sex organ that is producing the gamete are also physiologically and morphologically very much dissimilar. And the male sex organ we have discussed, that is the globule, and the female one is the nucleus. And this reproduction in Chara is mostly monoecious, which means that they are homothallic. Monoecious or homothallic means that the different sex organ, that is a male and the female sex organ, they are born in the same plant. This is the general rule in Chara, that most are monoecious, but certain dioecious species are also found. That is where the sex organs, different sex organs are born on different plant. So that rarity is Chara wallichi. In Chara wallichi, we see dioecious sex organ. I mean, this Chara wallichi is a dioecious plant of Chara, because in this Chara wallichi, the male and female organ are born in different Chara plants. This is the globule of Chara. We see that the globule of Chara is located in the nodal position of primary capitulum, just below the nucleus. This is the ellipsoidal nucleus of the female reproductive organ, and this is the globule or the male reproductive organ, which is globular in shape as well as very bright color. That is from orange, red to brown, and it is made up of 8 curved plate like cells called the shield cells. So it is mostly 8 shield cells that cover the globule, but sometimes it is also 4 in number, but that depend upon species. Here general rule is that it is made up of 8 shield cells, and each shield cell, they give rise to cylindrical plate-like shield cell, give rise to cylindrical manubrial cells. So this manubrium again bears cell called the primary capitulum in its terminal position, and this primary capitulum bears more cells called the secondary capitulum. So this is the manubrium. The rod-shaped manubrium give rise to primary capitulum, the primary capitulum which give rise to the secondary capitulum, and from each secondary capitulum arises 2 to 4 antheridial filaments. So these are the antheridial filaments that are born in the secondary capitulum, and each secondary capitulum bears 2 to 4 antheridial filament, and each antheridial filament, it give rise to has 25 to 250 cells. 250 cells, 25 to 250 cells in each antheridial filament. Now each antheridial filament, each cells of this antheridial filament, they give rise to antherozoids. So this antheridial filament, it is made up of chain of cells. This antheridial filament is made up of chain of cells, and from each cell arises the antherozoids. And antherozoids, how does it look like? It looks like it is a S-shaped structure, and it looks like an S-shaped structure. In the head region there are 2 flagella, and this is rostrum and also the nucleus. In the basal region there are the basal granules. So in each globule can give rise to at least 20,000 to 50,000 of these antherozoids. These are the antherozoids. These are the antherozoids, and each globule can give rise to 20,000 to 50,000 antherozoids. And now we come to the next slide. This is what I was talking about, the structure of globule and the antheridium. You can just see it is spherical in shape, red or orange in color, and it developed at the node of the primary lateral, that is, it is attached to the node by the pedicel cell, and it has 8 to 4 curved shield cells, and generally it is octoscultate, but sometimes it is also quadriscultate, that is in Chara zelanica. And from the shield cell arises the manubrium, which in turn gives rise to the head or the primary capitulum, and primary capitulum give rise to the secondary capitulum, and from secondary capitulum arises the spermatogenous filament or the antheridial filament, and from each antheridial filament arises a row of antheridial cell, and each antheridial cell give rise to a single antherozoid or spermatozoid. And each antheridial filament is made up of 25 to 250 cells, and we see that in each globule contain only 20 to 50,000 of antherozoids. And this is the structure of the each antherozoid. It has an anterior posterior region. Anterior region of the heterostella contains the flagella and the basal granule, and the posterior part, it contains the nucleus, and this whole structure, it is supported by microtubules. So this is the structure of nucleus, and that is the female reproductive organ, and it is ellipsoidal in shape. And this nucleus, it is green in color. Green in color, ellipsoidal in shape. It is made up of a basal cell, a basal. Here it is not that much written. It consists of a basal stalked cell and an egg cell surrounded by 5 tube cells. 5 tube cells that are spirally coiled clockwise, and they protect the Chara egg from environmental influence. And this tube cell, it ends in another 5 cell that is called the corona. So this is the corona cell. These are the tube cells. These are the tube cells. Inside the tube cells, that is the tube cell enmeshes the egg cell or the ovum. So this is all about the nucleus, which is placed in the nodal position just above the globule. In monoecious species, of course, certain species of Chara, they are dioecious. So those species, only the dioecious species, nucleus will be borne singly on a separate plant. So this is the all about the nucleus, that it is made up of, it is elliptical in shape, it is green in color, it is borne at the nodes of the primary lateral, the position. It is made up of one stalk cell, one egg cell, 5 tube cells that is spirally coiled, and top there is 5 corona cells or the crown cells. So this ovum, as it is laden with all starch and oils, and it has a receptive spot where the fusion occur. And this at the time of fertilization, this corona cell, they split, and certain gaps are produced, and through these gaps the antherozoid of Chara swims to the ovum. Now during the fertilization, the corona cell, gaps are produced inside in the corona cell, and during the time the shield cell, they gelatinize and exposing the antheridial filaments that are enmeshed in the gelatinous matrix. So the antheridial filaments, they are exposed by the desiccation or gelatinization of the tube cells. So when they are exposed to water, the wall of the antheridial filaments also gelatinizes, and the antherozoid or sperm are released. So these sperms, once released, they swim towards the nucleus by chemical attraction. Certain chemo attractant is released by the ovum or the egg cell. So they swim to the through the gaps in the corona cell, the gaps that are created in the corona cell, the sperm, the antherozoid or the sperm, they enter the nucleus, and they swim. And say if there is a certain there is a receptive spot, so they gather around this receptive spot, and one sperm or antherozoid fuses at the receptive spot. And once fusion is done, then the 2N zygote is formed. Zygote, that is 2N zygote is formed. And zygote, after a period of developing the cell wall and getting color in the walls, it is converted to a oospore which rests for a period of time, and it falls. This oospore inside the nucleus, entire structure, it falls to the ground and just rests for the period, I mean, till the conditions that are very conducive for the germination comes. So this is about the fertilization till the formation of oospore. So this is the theory per portion, that is, before fertilization the coronal cells they split, and the sperm liberated by gelatinization of the antheridial wall, and the sperm swim chemotactically then towards the ovum, and they swim inside the ovum through the splits in the coronal cell, and then they gather near the receptive spot, and one of the sperm or antherozoid fuses with the egg, and the zygote is formed. Once the zygote is formed, it secretes a wall around it, and then oospore is formed. So this is the germination in Chara. So the zygote, it forms four layers. The outer layer, that is it is formed of four layers. The outer layer, the outer two layer of the cells of the oospore, they are colored. The outer layers are colored, and the inner two layer, that is the inner two layer of the oospore, they are colorless. So this is the structure where the oospore, it is resting covered by the shield cell. So as the oospore germinates, the nucleus, it moves upward in direction, and then it divides mutically. Mutically, that is the reduction division into 4 nuclei. And after the formation of 4 nuclei, a lateral split is seen. A cell wall is formed where the upper lenticular cell has 1 nuclei, and the lower, the basal cell, it has 3 nuclei. Here we see the 3 nuclei. Out of this, only one survives and the later 2 degenerates. And as it degenerates, the basal cell contains only 1 nuclei. So the basal cell remain as it is. The upper lenticular cell, it undergoes a transverse oblique transverse division into 2 cells. The upper is the pyronemal initial, and the lower protonemal initial, and the lower is the rhizoidal initial. This rhizoidal initial, it gives rise to the rhizoid. The rhizoidal initial and the protonemal initial, it grows into 2 opposite directions. While the rhizoidal initial, it moves downward and produces many rhizoids, the protonemal initial, it moves upward and is divided into nodes and internodes. Here we see that the node is formed, and this is the internode in the protonemal initial, and this is the rhizoid, that is rhizoid developed from the rhizoidal initial. So in this way, the entire Chara plant is formed. This is how this germination, the theory of germination, that the oospore, it falls to the bottom of the pool, and it rests. Then when the conditions are conducive, that is, it gets water, it germinates. For the first step in germination is the the meiotic division, that is a reduction division into two cell, the upper small lenticular cell that forms the rhizoidal and protonemal initial, and the lower basal cell which contains three nuclei, two of which degenerate. It does not have any part in formation of the protonemal or in any initials. So we will be dealing with the upper lenticular cell that gives rise to the protonema. The protonemal initial that forms the primary protonema, and the rhizoidal initial that forms the multiple rhizoids. So from the primary protonema arises the secondary protonema, but that is for later course of time. So this is all about germination of Chara plant. So this was all about the sexual reproduction of Chara. I have already made videos about the vegetative reproduction in Chara. The link is in description box. I have also made videos regarding the thallus structure, the taxonomic position, and the economic, ecological, as well as the research importance of Chara. All the link is put in description box. Also, I have made videos regarding other algae. All the links are also in the description box. And if you want to make any suggestion, or want me to make any new videos, or discuss certain topic in detail, you can always write it in the comment section. So thank you for watching my video, and if you like it, do share it with your friends, and do subscribe to get the latest update of videos that I make. Thank you for watching.